Let's start by focusing on Corynebacterium, a genus of bacteria that plays an important role in both health and disease. These bacteria are rod-shaped with slightly rounded ends, and their structure is key to understanding how they behave and interact with the human body. Knowing their basic form helps us appreciate their biological functions and pathogenic potential. Now, what really defines Corynebacterium are several core traits. Many species naturally live on our skin and mucous membranes without causing harm, which is why they're part of our normal flora. They are gram-positive and have a distinctive club shape with granules inside. Importantly, they produce catalase, an enzyme that breaks down harmful hydrogen peroxide, helping them survive. They can grow with or without oxygen, showing flexibility, but they don't move or form spores. These traits together shape how they live and cause disease. Looking closer, Corynebacterium bacteria often arrange themselves in unique patterns, like V or Y shapes, which is quite characteristic. These arrangements, along with their staining properties that highlight granules inside their cells, help microbiologists identify them under the microscope. Understanding these features is crucial for accurate diagnosis and further study. When we compare different biotypes of Corynebacterium, we see clear differences in their virulence and appearance. The gravis biotype is the most virulent, producing fewer volutin granules and forming colonies that look like daisy heads. Intermedius shows moderate virulence with poor granule presence and frogs' egg-like colonies. Metis is the mildest, with the most granules and pouched egg-shaped colonies. Their hemolytic activity also varies, affecting how they interact with blood cells. These distinctions help us predict how dangerous a particular strain might be. The key to Corynebacterium's ability to cause disease, especially the diphtheria bacilli, lies in their virulence factors. Virulent strains produce a powerful exotoxin that can rapidly damage host tissues. This toxin is central to their disease-causing potential, allowing the bacteria to establish infection quickly and grow aggressively. On the other hand, virulent strains, which are common among people recovering from infection or carriers, don't produce this toxin and are less harmful. Let's talk about the diphtheria toxin itself. It's an exotoxin classified as an AB toxin, meaning it has two parts with different roles. Only bacteria carrying the tox gene produce this toxin, a result of lysogenic conversion. Fragment B is responsible for binding and entering host cells, while fragment A disrupts protein synthesis inside the cell. This disruption leads to the formation of a pseudomembrane, a hallmark of diphtheria infection. Understanding this toxin is crucial because it explains much of the disease's severity. Now, how does this toxin actually work? First, the receptor binding domain of the toxin attaches to a specific receptor on the host cell's surface. This binding is the crucial first step, allowing the toxin to enter the cell. Then, the toxin receptor complex is internalized by the cell through endocytosis, essentially engulfed by the cell membrane. This process sets the stage for the toxin to disrupt normal cell functions. Once inside the cell, the toxin undergoes important changes. The catalytic A subunit is cleaved but remains attached to fragment B by a disulfide bond. As the endosome vesicle acidifies, these bonds are reduced, releasing the catalytic A subunit into the cytoplasm. Meanwhile, the transmembrane domain helps this catalytic part cross the vesicle membrane. This release is critical because it allows the toxin. The final step is where the toxin exerts its deadly effect. The catalytic A subunit modifies elongation factor 2, EF2, by ADP ribosylation, which halts ribosomal protein synthesis. Without protein production, the cell can't survive and eventually dies. This mechanism explains how the toxin causes cellular damage and contributes to the severe symptoms of diphtheria, including tissue destruction and systemic effects. Understanding the progression of diphtheria infection helps us grasp its clinical impact. It begins when Corynebacterium diphtheria enters the respiratory tract. Soon, a pseudomembrane forms on the tonsils and pharynx, obstructing airways. The bacteria produce toxin that spreads systemically, leading to neurological symptoms and cardiac complications like myocarditis. This stepwise progression highlights why early detection and treatment are so vital. Diphtheria manifests in several ways. It's most common in children aged 2 to 10 and spreads through respiratory droplets, affecting areas like the facial, laryngeal, and nasal regions. It can also cause infections in other sites, such as conjunctiva, ears, vulvovaginal areas, or skin. Systemically, the toxin mainly targets the heart, adrenal glands, and nerves, leading to serious complications. Recognizing these manifestations is key for timely diagnosis. Symptoms of Corynebacterium infection fall into respiratory and cutaneous categories. Respiratory symptoms include sore throat, mild fever, hoarseness, pseudomembrane formation, airway obstruction, and systemic effects like myocarditis and meningitis. Cutaneous symptoms are generally milder, involving localized skin infections around the mouth and nose that usually resolve within 2 weeks. Knowing these symptoms guides clinical evaluation and management. Clinically, diphtheria is characterized by a pseudomembrane coating the tonsils and throat, which can be seen as a whitish or grayish layer obstructing airways. Another notable sign is the so-called bull neck, swelling due to enlarged lymph nodes in the neck region. These features, along with microscopic identification of the bacteria, confirm the diagnosis. Recognizing these signs early is critical for effective treatment. Laboratory diagnosis involves collecting 2 swabs from the lesion for staining and culture. Gram staining and Albert staining help visualize the bacteria, while cultures on Loeffler's serum slope and tellurite blood, agar, confirm their presence rapidly. The Schick test, an intradermal skin test, detects antitoxin antibodies, indicating immunity. These diagnostic steps are essential for confirming infection and guiding treatment. Testing virulence involves both in vivo and in vitro methods. In vivo, guinea pigs are used for subcutaneous and intracutaneous tests to observe reactions. In vitro, LX gel precipitation test detects toxin production by placing antitoxin impregnated filter paper on a gel with bacterial isolates. Precipitation lines form if the bacteria produce toxin, distinguishing toxigenic from non-toxigenic strains. This testing is vital for understanding the threat posed by specific isolates. Treatment centers on administering diphtheria antitoxin to neutralize circulating toxin, though serum sickness can be a side effect. Antibiotics like penicillins and erythromycin follow to eliminate the bacteria. Prevention relies heavily on vaccination, with a schedule starting at 6 weeks of age and including multiple doses and boosters through childhood. This combined approach effectively reduces. Finally, it's important to note the nomenclature. Corynebacterium is also known as Klebs or Loeffler's bacilli, named after Edwin Kleb and Friedrich Loeffler, who contributed to its discovery and study. Understanding these historical names helps connect modern knowledge with classic microbiology literature, rounding out our understanding of this significant bacterium.